Welcome back to the Deep Dive. Today, we're going to be taking a look at all the latest news in RNA sequencing.
Yeah, it really is exciting to see RNA sequencing going from something that was just a research tool, in a lab, to something that is now actually affecting people's lives and having real-world impact.
It's really amazing. And, it seems like every day there's a new headline about RNA sequencing. So we thought, it was time for a deep dive to kind of see what's going on.
Yeah, there's a lot to cover for sure. I mean, we've got stories today about brain cell development,
Oh,
fish farming, and even AI that can write DNA on demand.
I mean, that sounds like science fiction.
It does, it really does.
Where do we even begin?
Well, I thought we could start with the brain cell development story.
Okay.
Researchers at the Southwest Research Institute have developed this really cool new computational method that allows them to use RNA sequencing data to track brain cell development over time.
So they can actually see how brain cells are developing and changing.
Exactly. It's like, imagine being able to watch a brain as it's being built
Whoa.
and see how all those connections are forming and, how these cells are becoming specialized. It's really incredible.
Yeah, that's pretty mind-blowing. But why is this so important? I mean, why do we need to track brain cell development?
Well, I think it's important for a couple of reasons. First, it could help us understand how neurodevelopmental disorders develop, like, autism, for example.
Yeah.
By seeing how brain cells develop in a typical brain versus a brain with autism, we might be able to identify some of the early changes that lead to the disorder and then potentially develop new therapies.
That makes sense.
And second, it could help us develop new treatments for brain injuries and neurodegenerative diseases like Alzheimer's or Parkinson's.
Yeah, I mean, if we can understand how brain cells develop, then maybe we can figure out how to regenerate them.
Right, exactly. And that's, that would be huge. That would be absolutely revolutionary. I mean, if we could repair damaged brain tissue, it could change the lives of millions of people.
I mean, it's almost like we're talking about science fiction.
It is. It's very sci-fi, but it's based on real science. And that's what's so exciting about RNA sequencing. It's giving us these tools to do things that we never thought possible before.
Yeah, so that's brain cell development. What's the next headline?
So the next one is actually kind of an interesting pairing. Researchers have used single-cell RNA sequencing to compare horse and human pregnancies.
Horse and human pregnancy.
Yeah, I know it seems a little strange, right?
Yeah, I don't really see the connection there.
Well, horses have actually been used as models for human pregnancy for a long time.
Oh, really?
Yeah, because they have a lot of similarities in their reproductive systems, but this new research actually takes it a step further. By using single-cell RNA sequencing, they're able to look at the individual cells involved in pregnancy and compare them between horses and humans.
So they're able to see how the cells are interacting with each other and how they're changing over time.
Exactly. And it's really amazing what they're finding. They're seeing all these parallels between horse and human pregnancies that we never knew existed before.
So even though horses and humans are very different species, at the cellular level, there are a lot of similarities.
Yeah, that's right. And that's what makes this research so valuable. It's giving us a much deeper understanding of pregnancy and how it works, which could lead to new ways to prevent and treat pregnancy complications.
Wow. So that's horses and humans.
Horses and humans. So the next one is about sugar beets.
Sugar beets. Okay, I'm intrigued.
So this one is a little bit more down to earth, but still really important. So scientists are using RNA sequencing and genome-wide association studies to identify genes that control sugar content and biomass in sugar beets.
So are they trying to make sugar beets sweeter?
Well, yeah, that's part of it. I mean, sweeter sugar beets mean more sugar, and that's important for food production and also for biofuels.
Right, because sugar beets can be used to make ethanol.
Exactly, but it's not just about sweetness. They're also looking at biomass, which is the total amount of plant material.
Okay.
And if they can increase biomass, that means more sugar beets per acre, which again is good for food production and biofuels.
So this research could have a big impact on the environment and the economy.
Absolutely. I mean, sugar beets are already a major crop worldwide, but if we can make them even more productive and sustainable, it could make a big difference.
That's pretty cool. So we've got brain cell development, horse and human pregnancies, and sugar beets. What's the last headline?
This one is about personalized medicine. Foundation Medicine and Sumitomo Pharma are using RNA sequencing to develop a companion diagnostic for acute leukemia treatment.
Okay, companion diagnostic. Break that down for me.
So a companion diagnostic is a test that helps doctors choose the right treatment for a patient. It basically tells them whether a particular drug is likely to work for that patient or not, based on their genetic makeup.
Oh, okay. So in this case, they're using RNA sequencing to identify specific genetic mutations in leukemia patients. That might make them more or less likely to respond to certain treatments.
Exactly. And this is really important because it could help doctors avoid giving patients treatments that are unlikely to work, which can save time and money. and also prevent unnecessary side effects.
So it's all about making treatment more personalized and targeted.
Exactly. And that's really the future of medicine. We're moving away from this one-size-fits-all approach to an approach where treatments are tailored to the individual patient based on their unique genetic profile.
And RNA sequencing is making that possible.
It really is. It's giving us this incredible insight into the inner workings of cells, and that's revolutionizing healthcare in so many ways.
Yeah, it's pretty amazing. So we've got RNA sequencing being used to study everything from brain cells to sugar beets to cancer cells.
And that's just the tip of the iceberg. I mean, there are so many other applications for RNA sequencing. We could talk about this all day.
Well, I definitely want to hear more, but I think we've covered a lot of ground already. Maybe we should take a break and come back to this in part 2.
Yeah, that sounds good. We can delve into some of the other fascinator applications of RNA sequencing and maybe even talk about some of the ethical considerations.
Yeah, because this is powerful technology, and it's important to think about the implications.
Absolutely. So stay tuned for part 2 of our deep dive into RNA sequencing, where we'll explore even more groundbreaking discoveries and their potential impact on the world. Welcome back to the deep dive. So where were we? Yes, RNA sequencing. It's really shaking things up, huh?
It really is. We were just talking about how it's being used in everything from brain cell research to personalized medicine.
Exactly. It's incredibly versatile. But, with great power comes great responsibility, right?
That's true. I mean, some of these technologies are so advanced, it makes you wonder about the ethical implications.
You're hitting the nail on the head. I mean, take AI-designed DNA, for example. We've talked about its potential, but we need to tread carefully there.
It's like opening Pandora's box. We don't fully understand the complexities of biological systems.
Right. Who knows what unintended consequences could arise? It's not as simple as just plugging in some code and getting a desired outcome.
Exactly. We could accidentally create something harmful or disrupt delicate ecosystems.
It's a sobering thought. And then there's the potential for misuse. This technology could fall into the wrong hands.
That's a scary thought. Biosecurity needs to be a top priority. We need to make sure this technology is used for good.
Couldn't agree more. We need scientists, ethicists, policymakers, everyone working together.
It's a societal issue as much as a scientific one. We can't just bury our heads in the sand.
Right. Open and honest conversations are crucial. We need to figure out the boundaries before things go too far.
Okay, enough doom and gloom for now. Let's get back to some cool RNA sequencing discoveries. There was one about RNA stability that caught my eye.
Yes, the half-pipe tool. This is a really neat development. You know how RNA molecules carry genetic information?
Right, like messages within our cells.
Exactly. Well, RNA stability refers to how long these messages last before they break down. It's like how long can they deliver their instructions before they disappear?
Makes sense. So half-pipe helps scientists measure how long those messages stick around.
Exactly. It gives them a precise measurement of RNA half-lives. This is super important for understanding how genes are regulated.
Oh, I see. Because if the message disappears too quickly, it can't do its job.
Precisely. Imagine trying to follow a recipe where half the instructions fade away before you can read them. Chaos.
That's a great analogy. So what did the researchers actually do with half-pipe?
They studied RNA stability across a whole bunch of organisms. Yeast, mice, even zebrafish.
Zebrafish, those tiny see-through fish.
Yep. They're amazing for research because you can actually see what's happening inside them. The researchers learned a ton about how RNA stability impacts development.
So cool. Those tiny fish are helping us unlock big secrets.
Absolutely. Nature is full of surprises, and RNA sequencing is helping us decode it all. Speaking of decoding, remember those other applications we touched on?
You mean the sugar beet research and the personalized medicine for leukemia?
Exactly. Oh, and there was one more I wanted to mention. It's about how RNA sequencing is helping us class about cells.
Cell classification. What's that all about?
Well, imagine you have a giant box of Legos, all different shapes and colors. You want to organize them, right?
Yeah, so you can find the right piece when you need it.
Exactly. That's what cell classification is like, except instead of Legos, it's cells. And with single-cell RNA sequencing, we can analyze the gene expression of each individual cell.
So instead of looking at a whole tissue, they're zooming in on each cell.
Exactly. This gives us a much more detailed picture of what each cell is doing and how it's different from other cells.
I can see how that would be valuable.
It's like discovering hidden rooms in a giant mansion. We're finding all sorts of new cell types and subtypes that we never knew existed.
It's amazing how much we're learning about the human body.
And about life in general. I mean, we've talked about brain cells, sugar beets, fish, even individual cells. RNA sequencing is everywhere.
Yeah, it's really revolutionizing biology.
It really is, and I feel like we're just getting started.
It's mind-blowing. So much information to process. Maybe it's a good time for a quick breather before we dive into the final part of our deep dive.
Sounds good. We'll be back in a flash to talk about the future of medicine and beyond.
And we're back for the final part of our deep dive into RNA sequencing. You know, we've talked about all the amazing research that's happening, but what really gets me excited is seeing how this technology is actually being used to help people.
Yeah, it's really starting to move from the lab into the real world.
Exactly. And one of the areas where I think it has the most potential is in personalized medicine. The idea of tailoring treatments to each individual patient.
Absolutely. That's kind of the holy grail of medicine, right? To be able to treat each patient as an individual rather than using a one-size-fits-all approach. And RNA sequencing is making that possible. I mean, by analyzing a patient's RNA profile, doctors can gain all sorts of insights into their disease. They can predict how they're going to respond to treatment and even identify a new drug target.
It's like having a personalized roadmap for each patient's healthcare journey.
Exactly. And as the technology becomes more affordable and accessible, we're going to see this happening more and more. I mean, imagine a world where every patient gets a treatment plan that's specifically designed for them.
Yeah, that would be incredible. Fewer side effects, better outcomes. It would really revolutionize healthcare.
It really would. And it's not just about treating diseases that already exist. RNA sequencing is also helping us develop new drugs. By analyzing the RNA profiles of cells and tissues, scientists can identify new drug targets and then develop therapies that are more effective and have fewer side effects.
I mean, it's like having a secret decoder ring for the language of life, right?
Exactly. We're finally starting to understand how diseases work at the molecular level. And that's giving us the tools to develop truly targeted therapies.
And it's not just limited to human health. I mean, we talked about the sugar beet research. RNA sequencing is also being used to develop new crops that are more resistant to disease and drought, which is hugely important for feeding a growing population.
Absolutely. I mean, RNA sequencing is helping us tackle some of the biggest challenges facing humanity, like climate change and food security. It's really pretty amazing.
Yeah, it is. It really shows the power of science to make a positive impact on the world.
And we're just getting started. I mean, the field of RNA sequencing is constantly evolving. New discoveries are being made all the time. It's a really exciting time to be involved in this field.
Absolutely. It's been an incredible journey exploring the world of RNA sequencing with you.
Yeah, it's been a lot of fun.
We've learned so much about the incredible potential of this technology, from brain cell development to personalized medicine to sustainable agriculture. It's clear that RNA sequencing is going to have a profound impact on our world. And it's an impact that we need to think carefully about. With great power comes great responsibility.
Couldn't agree more. We need to make sure that these technologies are used for good and that they benefit everyone.
Right. It's up to all of us to shape the future of science and to ensure that it's a future that we can all be proud of.
I am optimistic. I think we're headed in the right direction.
Yeah, me too. And I think RNA sequencing is going to play a big role in getting us there. So to all of our listeners out there, stay curious, stay informed, and keep exploring the wonders of science. And that's it for this week's deep dive into the world of RNA sequencing. Thanks for listening.